difference between X-linked dominant, X-linked recessive, and then I'll do this pedigree. So let's see how this would make sense. If there is a gene located on the X chromosome, okay, if the disease gene, let's say we'll talk about X-linked recessive first. X-linked recessive first. If the disease gene is located on the dad's X chromosome, in this case the dad has only one X chromosome. If it's recessive, that's all you have. You don't have two copies, you have only one. So the dad is automatically diseased. In X-linked recessive diseases, when a diseased father marries a healthy female, no children will have a disease. In this case, assuming both genes of the female are normal. When a diseased father marries genotypically homozygous normal female, all the children will have normal phenotypes. For example, in this case, sons will never have a disease because the sons will get the X chromosome from who? From mom. In mom, both copies are normal. Okay, so this is one possibility. Again, this is not the only possibility. Okay, in this case, all sons will be normal, genotypically and phenotypically. But all the daughters will be carriers. You're just spreading the gene, you're spreading the disease gene in the population. So when they marry, so all the daughters will be what? Carriers. So when these daughters marry a healthy, so now of course, let's say in this case, the next case scenario is that, are we okay with this so far? Okay. So let's say these daughters marry a healthy male. They marry a healthy male. But all the daughters are what? They were carriers, right? But they are carriers. Now when there's a carrier female, which is phenotypically normal, because you cannot recognize these female whether they have the disease gene or not, unless you get a court's order and get their genotype, right? You wouldn't know. Male, either they're diseased or not. It's on their face. You will see them right away. If they're diseased, they'll express it. If they're not, they will not. So now a healthy male marries a carrier female, which means
50% of males will be diseased.
Yeah. Daughters will never have a disease. Daughters will never have a disease. Okay? Daughters will never have a disease. Why? Because they're getting the healthy chromosome from father. When it comes to X-linked recessive, male-to-male transmission is not allowed. True? Female-to-female transmission is allowed, but for any female to have a disease, dad must be diseased. This is a requirement. In X-linked recessive disease, no daughter can have a disease unless the father is diseased, minimum. And the mom must be a carrier, minimum. It's almost like autosomal recessive disease for female, because they have two copies of X chromosomes.
Again, here with this case, nobody will be diseased.
No, the son will be.
The son, have 50%.
Which one?
The son. The first one. YX.
This one.
This one will be diseased?
Yeah, because he got an X chromosome that is diseased. He gets only one X chromosome, and that's diseased. So he will have disease.
So 25.
The reason daughter will not have a disease, look, the reason this daughter has no disease, she has one abnormal copy, one healthy copy. Because female gets two X chromosomes, the likelihood of them getting both chromosome disease is very rare. They usually will get one normal, one abnormal, so they won't express the disease. So all X-linked recessive diseases are common in who? In men. Always in men. Always in men. I think the ratio is 90 to 10%. Rarely would a female have X-linked recessive disease. Even the 10% is extremely rare. So for a female to have a disease, father must be diseased, mom, minimum, has to be a carrier.
So the father can't be carrier?
No. Because
He can't be carrier.
no. Yeah.
He should be diseased or
Always diseased or not diseased. Yeah. Okay. Alright, so X-linked dominant is the same thing as autosomal dominant. X-linked dominant diseases don't exist that much. Autosomal dominant diseases are rare. X-linked dominant is even more rare. Okay. The only X-linked dominant disease that I can think of is rickets. And there's two types of rickets. This is actually vitamin D resistant rickets, which is X-linked dominant. Okay. Again, rickets is calcium deficiency, right? Or hypocalcemia due to vitamin D deficiency. But in this case, it would be genetic receptor deficiency. So let's go over something. You tell me, because once you practice this will help you the most. Whatever I talked about will make you help you understand. You tell me, what is this condition?
What does that mean?
Green means disease.
Autosomal recessive.
Green means disease. If you don't like green, I'll make it red. That means dangerous, right? On my thing, you see the green area is close to me. It's big, so I was...
So is the circle male or female?
Autosomal dominant.
Okay, autosomal dominant was one choice.
The circle is a female?
Oh, I have to talk about it? Okay, let me tell you.
Yeah, of course.
Let's go with pedigree first, okay? Let's go with pedigree. What are the regulation of pedigree? Circle always means female. Rules of pedigrees. Circle means female. Square means male. Okay. Shaded circle or means affected. Shaded means affected. Carriers are usually not shaded. If it's half shaded, you'll be lucky. That means carrier. Okay, so half means carrier. Complete shaded means affected. In this case, diseased. So number one, circle, female. Square, male. Complete shaded, square or circle, whichever one, means affected. Partially shaded, half, means carrier. Non-shaded means normal. Okay, means normal. But again, it depends. Not everybody will give you half a shaded. So sometimes you don't know if it's a carrier or not. We'll do some example. When two people are connected by a straight line, horizontal line, it means marriage. Horizontal line means marriage. Horizontal line means marriage. Vertical line means children. Vertical line means children. Now, if it's one son, it'll be just one square. But if it's multiple, then what do? Relatives are connected by what? By horizontal line, connected by verticals. That means children. In this case, 3 daughters, 1 son. Okay? Horizontal line between male and female means marriage. Now, this will not apply to homosexuals because they cannot have children. So if there's a horizontal line between them, yeah, they're married, but there will be nothing beyond that. Okay? So, a vertical line then connecting to a horizontal line that has multiple children should be connected by vertical lines further. If it was one child, it would be just here. But because it's multiple children, then that means, in this case, whenever you have vertical lines connected by horizontal lines, all of them, that means siblings. Let me show you. This means sibling. This. Green. This purple means marriage. This blue means one source, one family, one parenthood. All of them come from a single, 2 parents. Okay, all from the same parent. Single line. But let's say we have something else. Here is, here's a female. Here is a male. Now, if the origin is the same, like this, that means twins. 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 Okay? Twins. You see here, the 2 children are separated by a horizontal gap. You see this? Means time. Let me color it red. This means time. That means they're not twins. Time and time. But here there is no time. It's at the same time. This is dizygotic twins. But let's do it something different. If you have a straight line, but yet this one splits, this is monozygotic twins, identical twins. Okay, identical twins. I don't think you'll ever see twins and stuff on the exam, but as long as you know what is marriage and what are children's. How do you know what's children of what, who's children of male, female, children, and affected, non-affected carrier? That's what's important. What if I do this? In some cases, what does this mean? You don't know the gender of this child. Gender of this child. We don't know if it's a male or female. I don't want to tell you. Okay. Can I add it here? Sure. Female, female, male, female, don't know yet. Okay, maybe the ultrasound is undetermined yet, or this is still a fetus, or whatever. Anyways, okay, so know these basic things. This should help you. Okay, now what do you think? Autosomal dominant. So one options, whatever you think, tell me because it will help us. Choice A, autosomal dominant. Anybody wants to have choice B? Mitochondrial. Anybody wants to have choice C? Okay. X-linked recessive.
Oh, no.
Okay. So,
Because the father should be...
yeah. So look, let's look at this. Mother has a disease, right? All the children, daughters and sons, they both have disease. This cannot be X-linked. In X-linked, remember, mom will only give a disease to who? To their son. Unless it's X-linked dominant. Okay, this can be X-linked dominant so far. But look, this female, she's affected. And she has all her children again have a disease. This father has a disease. None of her children has a disease. So this has to be mitochondria. You only get it from who? From mom. You never get it from dad. Okay? If it was autosomal dominant, every generation will have it, so the father should at least have one son. If it was X-linked dominant, at least one of the kids, in this case, at least one of the kids, especially the daughter, would have a disease. The only difference with the X-linked dominant is that if a father has a disease, again, sons will never have a disease. Rule number one for X-linked diseases, male-to-male transmission is not allowed. Male-to-male transmission is not allowed. So both daughters should have been diseased. All I have to do is change it this way. Let's see if I can change this. Oh, that's not good. Now I'm stuck with this, huh? Let's see if this works. This is one thing I didn't want to do. Oh my god. I have no choice but to make everything red because otherwise it will not make sense. I don't want to draw a new one from scratch. Okay. Now let's go with this. What if, now the dark one means disease, okay? What do you think now? So tell me about autos. I will ask you choices. You answer me. X-linked recessive. Can normal parents have a diseased child?
Yeah.
Yes. Normal parents can have a child, right? How about autosomal recessive? They skip generation, right? So do you see skip generation? No. So this is not recessive. This has to be dominant. So number one, when you see no skip generation, automatically you should assume this is a dominant inherited disease. Now, one more thing I probably didn't tell you. Generation, this is one generation. This is the second generation. This is the third generation. Does it make sense? Yeah. Each generation has a disease, so this has to be dominant. So we at least know 50% of the answer. When there's no skip generation, every generation has a disease, it has to be dominant. But we don't know if this is X-linked or autosomal. Let's see if you can help me with that. What makes it X-linked? What makes it dominant? Or is it the mitochondria? It's not mitochondria. Why is it X-linked?
Because the father didn't inherit his son.
So here, this is generation 2, right? Individual 2, true? So individual goes 1, 2, 3, 4 from left to right. Generation 2, individual 2 has a disease, and he's a male. He was able to give that gene to his daughters, but not to his son. So this has to be at least X-linked. So look, each generation, the mom can give it to son or daughter. X from mom can go to daughter and son. Same thing here. So mom gave it to her son, but this dad cannot give it. So, but he was able to give it to her daughter. So this is dominant, but X-linked. Okay. Now what do you think? It is recessive. Dominant or recessive, so it's recessive. X-linked or autosomal, what do you think? Why autosomal?
Nobody had it until
Only a female has it. So that means if this was X-linked, you know, the mom and dad, for a daughter to have a disease, recessive disease, the dad must be diseased. You have to get one copy from each parent. So this is a daughter, female. Female must have both X chromosome disease. True? So at least one of the X chromosome has to come from dad. If dad has a disease X chromosome, and he is not dizygotic, he's hemizygous, right? So he must express the disease. Since the dad is healthy, that means his X chromosome is normal. So this is not X-linked. Make sense? Okay. Okay, let's see what else. So we talked about dominant, X-linked dominant.
What is the answer was for this one?
Autosomal recessive. Autosomal recessive. What do you think now? You're asking me, or you're sure? Let's work through this. Look, generation, individual 1, individual 2. They got married. And they had 4 children, true? 3, 4, 5. In the generation 2, individual 2, 3, 4, 5s are all siblings, correct? And they are children of 1 and 2. So only one of them have the disease. Individual 3 in generation 2. And that's a male. He's a male. Can a male get a disease from his mom or dad if it's X-linked?
Yeah.
Only from mom. So yeah, this could be recessive so far, or X-linked, recessive or autosomal recessive. But let's focus on 1 and 2. If this was dominant disease, normal parents can never give you abnormal child. It has to be recessive. The only disease where you have normal parents and they can give you a diseased son or daughter is what? Recessive. So automatically you have to make this conclusion. And there is skip generation, of course. So if it's skip generation, what are you going to think? Recessive. This generation is normal, right? And this generation has a disease. But look at this generation. Normal. And this generation has a disease. So don't include this as these two. You say, oh no, skip generation. There is skip generation. Okay, because these parents don't have a disease. Yeah. Okay, so number 1, whenever you have normal parents giving an abnormal child, it has to be recessive. You can never have a dominant disease from normal parents. Think about it. You cannot have a disease from normal parents if it's dominant. Each generation must express the disease. Dominant, you need only 1 copy, right? If you have that 1 copy, you better express it. Otherwise, it's not dominant. Dominant disease, for it to be disease, you need 1 copy. You either have it or you don't. You can't hide it. So if you see parents that are normal, they don't express the disease, you don't think this is dominant. This cannot be dominant. Either they have it or you don't. There's no carrier state in dominant. There's no carrier state. The carrier state is for healthy, not for disease. Okay? So this has to be recessive. Now let's think about it. Only males are getting the disease. So it has to be X-linked. Only men are getting the disease. It has to be X-linked. Okay? Only men are getting the disease. Huh? I didn't hear you. I think this thing is too loud.
That's so be the probability.
Probability?
Yeah.
Like whether you get it or not?
No, I mean like just the fact that you don't know to be a coincidence.
No. Let's see. Let's see. Let's see how that would work. Now, probability means what? Most likely. And when we talk about these things, what is most likely? You know, whatever is most likely is factual these days. Okay? The only way this kid, male, can have a disease is if you get 1 copy. If it's autosomal, you can get 1 copy from this. Let's say this is big A. 1 from each. So it's possible. Right? It is possible. Now, this is also possible. Okay? That's also 25% makes sense here. But there's the thing. When you have this scenario, this person came outside of the family. It's not part of this family. This marriage. So whenever you think about these diseases, again, you should be assuming that this is what? Normal. If you think about the autosomal. So in all likelihood, yes, there is a chance. And that chance is probably 1%. Compared to X-linked, 99%. So what would you go with? Of course, yeah, the higher probability. So it is possible, but whenever you see only men getting the disease, and all the females are normal, and men can never get a disease from their father, because fathers are normal, then this is unlikely to be autosomal. It has to be X-linked. Yeah. Okay. So you give it your best chance. Of course, in all likelihood, there's crazy things that happens. Yeah. Yeah.
Let's just say we refilled in one of the abdominal circles. That would have changed it to.
Give me which individual. Give me the hypothetical case. Which individual would have a disease?
Only the one to the right.
Well, give me the numbers.
Second generation.
Second generation individual 5?
Yeah.
Okay, good. Now you're talking to me, see? This one, right? Okay. If this person has a disease,
Would that now make it autosomal?
no, no, not necessarily, not necessarily. Okay. You can think about it, but still, you know, if this was the case, one of the daughter would. I will show you at least one of the daughter. Whenever you have a pedigree, there's a possibility there. When you see a pedigree, clear cut, only male are affected, think of X-linked. Just to make it easier.
That's where I was going. That's why I said one of the, let's say, daughters from second or third generation were filled in, and they were affected, and that now changed to autosomal recessive.
And
Because you started with two normal autosomal.
yeah, so in this case, if she's part of the family, yeah, this has to be. Otherwise, if this daughter could not have got this disease unless this father was not diseased, if it was X-linked. The only way daughters can get a disease, X-linked recessive disease, if the father must be diseased. Where did she get the second gene from if it was autosomal? From the father. So this will 100% make it autosomal recessive because this female must have 2 copies of the recessive gene. One from dad, one from mom. So let's say she got one from mom. Since the mom is normal, it has to be heterozygous. The dad is normal, he has to be heterozygous. And this can no longer be X-linked, yeah. Yeah.
So would that still be skip generation? Because the next generation right after that has the disease or no?
In this case, this is normal, so it's skip generation, yeah. As long as you have one generation, it's skip generation. It doesn't have to be always alternative generation. Yeah.
Also, the first individuals of the second and third generation don't have.
Yeah. The second and third generation, the first individual, this, and this.
Yeah, so doesn't it also count as skipping generations if any other children?
Yeah, you can think about it that way, yeah. But not all children will have the disease. So as long as one member of the family has a disease. Yeah.
There's in that generation.
Amar, you had a question? Never mind? Okay, I won't mind.
So, sorry, Mr. Park, you said it's still it's autosomal recessive, right?
No, when you add this female,
Yeah.
this person, the one I'm circling, okay? For her to have a disease, because whether she's X-linked or whatever, this person must get both copies from, one from each parent. The only way you can get a disease gene from your dad is if he's autosomal. If he was X-linked, the female he must have expressed the disease. He must have been shaded.
And that would all have it.
Yeah. And this male will not have it, exactly. Yeah. Okay. Any other? Okay, so let me give you one more example of pedigrees. Uh-oh. This is a problem. Huh. I just don't want to draw too much.
Make it all easy.
Yeah, let's
We
see.
didn't square right again.
We have a message set, so we put squares in.
There you go. Okay, let's do one more to see how that would work. Hopefully I can remember. Okay, do you see? I put carrier, right? Can you guys see that? So let's just make the mom, I guess, because it's easier. Carrier. And then here, not from the outside, but from the family. Let's make this dad a carrier, and this mom a carrier. Are we good? They're brother and sister, right? one brother and one sister, they're carriers. And then when the mom is a carrier, they will have a son with disease or a daughter. And when the dad is none. What do you think now?
Auto recessive. Auto recessive.
Autosomal recessive.
Remember, for X-linked, there is no paternal carrier state.
Right. So auto recessive.
No, but then there's a dad who's carrying.
Yeah, it's auto recessive. Yeah, no, auto recessive, so that's why.
It's healthy mom and dad, so it can be.
And they skip one generation.
Well, this is based on, look, so you see the dad has a carrier state,
Yes.
and then all of his children are normal. When the mom has a carrier state for the same gene, one of the child has a disease. So that means you get the disease from mom, but not from dad. Both have abnormal gene. And this can be vice versa. Okay, so there's two cases in this case. One disease known as Prader, Willie. This is imprinting, and then Angelman syndrome. So two conditions. Okay. Now, depending who has the deletion, both of them chromosome number 15. Chromosome number 15. If the dad has chromosome 15 deletion, or mom has chromosome 15 deletion. One chromosome, not both. They should have, each of them should have what? 2 copies of chromosome 15, correct? Both parents would have, every human being is diploid. They must have 2 copies of each chromosome. Mom should have 2 chromosome 15, dad should have 2 chromosome number 15. If chromosome 15 is deleted, it's not entire chromosome, just a piece of it, the gene. And the gene is known as Prader-Willi gene, and here it's known as the Angelman gene. So on chromosome 15, paternal chromosome 15, if the gene is deleted, okay, let's say the son or one of the child inherits this. I'll do it this way. Let's just do it this way.
So Prader-Willi is from paternal.
Very good. Deletion from paternal. So let's say if one of the child gets inherit the deleted chromosome from dad, and in this family you inherit the deleted gene from who? From mom. Okay? Now additionally, let's say you inherit the normal gene from here. Okay? And then you inherit the normal gene from here. The children are again carriers. They wouldn't have any disease unless chromosome I'm going to cover in red. This chromosome is inactivated randomly, and this chromosome is inactivated randomly. So in imprinting, you must get one deleted copy from one parent, and then the normal copy that you get from the other parent is inactivated. At the end, you have 0 product. That's called imprinting. So it's just like autosomal recessive, because chromosome 15, but in addition to that, the normal copy is inactivated. So it depends whether you get the deleted copy from mom or dad. It tells you if it's Angelman or Prader-Willi's. If you inherit the deleted copy from dad, it's Prader-Willi's disease. If you inherit the deleted genes from mom, Angelman syndrome. Okay? So you must, first you must inherit the deleted gene from one of the parents. And then you must inherit a normal copy of the same chromosome from another parent. So the normal copy is inactivated randomly. If it's inactivated, it's called imprinting. If it's not, then nothing, no problem. Let me do it in another way. It could be confusing if you don't take your time and think about it. So we have mom and dad. Mom has chromosome 15, 2 copies. Dad has chromosome 15, 2 copies. Good? And then mom has in one of the chromosome 15 a deletion. Good? Okay. Now, remember, chromosome segregation is independent event. So you don't know which chromosome you'll get from mom. It's 50-50 chance. Let's say you get the disease. One son gets, again, there's 4 possibilities, right? Child 1, so the possibility of children are 4. Let's say 1, 2, 3, 4. Always there will be 4 possibilities. So one child gets this chromosome from mom, and the second child gets this chromosome from mom. In this case, I guess this is not much, so I'll just keep it 2. And this guy gets this chromosome, and then this gets this chromosome. Let's erase these guys. So look, 2 children. One child gets a normal copy from mom, and one child gets abnormal copy from mom. Both paternal copies are normal, no big deal. Blue is normal. Is that okay? Okay, blue is normal, so the dad chromosome is normal. So which one you get, it really doesn't make a difference. But from mom, there's one normal copy and one abnormal copy. So in this case, you still have 50% of the gene, not a problem. So this person is normal and this person is normal. Good? Are we good so far? Now there's something called epigenetic. Epigenetic. Afterwards, once this child inherits this copy, the blue copy is inactivated randomly. This is inactivated. Or specifically that area of the gene, not the entire chromosome. This area is inactivated. So there was 2 things. One was deletion. And then second thing, there was inactivation. 2 things occurred. So it depends if you get the deleted copy from mom or dad. And then the second gene that you get from the mom or dad is inactivated. So what you're going to think about is if paternal copy is deleted and maternal is inactivated, it's a different thing. If maternal copy is deleted and paternal is inactivated, that's a different disease. So at the end it gives you 2 different diseases. It's the same chromosome, chromosome 15. It's famous for that. That's called imprinting. And the inactivation is called methylation. It's methylation that causes the inactivation. I'll do it one more time, hopefully it helps you. Chromosome 15 is a big chromosome. So mom has chromosome 15 and dad has chromosome 15. Let's say on mom, chromosome 15, the maternal chromosome 15, this gene is deleted. Okay? The gene is deleted. Now, if you get one from mom, one from dad. Let's say from mom you get the disease gene, and from dad you get the healthy gene. Both are chromosome 15. The child has no problem so far, because at least they have one gene that's active and the other gene that's deleted. But for whatever reason, this gene that is here, that is normal, green, is now inactivated. At the end you have no product. It's almost like both genes are deleted. So if the deletion is from mom, it's Angelman. If the deletion is from dad, it's Prader-Willi. Does it make sense? Okay. So number 1, in imprinting, there is deletion involved of one allele, and the second allele is inactivated. Okay, so focus on that. In imprinting, one gene, one copy, so each gene must have 2 copies. If the first copy is deleted, the second must be inactivated. If you don't inactivate it, it's normal. So there is deletion and inactivation of the same gene. But the same gene has 2 copies. In imprinting, if one copy is deleted and the second copy is inactivated, it's called imprinting. Imprinting can give you 2 possibilities, 2 diseases of the same chromosome. It depends if the imprinting, if the deletion is on the dad's gene and activation on the mom gene, it's called Prader-Willi's. Let's flip it now. If the deletion is on the mom's gene and the inactivation is dad's gene, it's known as Angelman. So, but for you to remember, focus on deletion. If paternal gene is deleted, then it's Prader-Willi. If maternal gene is deleted, it's Angelman. Okay? Look, the second copy must be inactivated. If it's not inactivated, it's not a disease. It must be inactivated.
What usually causes that?
Hmm?
What usually causes that?
Environment, methylation. So it's the process. So randomly it's methylated. Okay, not everybody will get it. Angelman's patients are known as happy puppies. These children are always smiling. If you during vaccination, guess what they do? They don't cry. They smile. They do feel pain, but they don't know how to express it. They're always obese, they're floppy, and they're happy. Prader-Willi, they're a little bit angry. They're the opposite. Yeah. They can have self-mutilation, they can have those issues. Angelman is called Angelman. Why? Because angels supposed to be happy, they don't feel any pain, and they're like relaxed all the time. That's what, if you will rarely see them, but there was a case, I think, 2 years ago in St. Anthony Hospital, I seen one. So, they're always smiling. They're so happy. It's crazy. But again, your focus is on what? On deletion, deletion. If the deletion of... we're only talking about which chromosome? 15. If chromosome 15 is deleted, or part of the gene is deleted, then it's Prader-Willi. If chromosome 15 on maternal side is deleted, then it's Angelman. The second copy of the same chromosome must be inactivated, otherwise it's normal. So maternal deleted, paternal inactivated, it is Angelman. Paternal deleted, maternal inactivated, Prader-Willi. Okay? You had a question, then I'll come to you. Said, you had a question?
Oh, yes. What happens if the child inherits both deleted chromosomes?
It's autosomal recessive.
But is it the same disease?
Yeah. No, it's something else. It's slightly different because inactivated gene has still some product. So, but it's rare. Prader-Willi coming from complete deletion is rare, but it will be called autosomal recessive. Yeah. If one copy deleted gives you a disease, it's autosomal dominant because chromosome 15 is autosome. It's not X or sex chromosome. If one copy deleted gives you a disease, so let's do this. Let me give you some varieties. In this case, look, one copy, so this is 15 from mom and this is 15 from dad, let's say. One copy deleted, one normal. No disease, this is what? Autosomal recessive, right? If there's no disease, it's normal. But for autosomal recessive, both copies must be deleted. If one copy is deleted and the other one is normal and you get a disease or symptoms, it's autosomal dominant. Okay, so this is autosomal recessive disease, but the second copy is inactivated rather than deleted as well. So it's still autosomal recessive, but it's called imprinting in a way because the mechanism is different.
So inactivated genes, they still produce some product.
If you activate it from time to time. But in this case, during the childhood, they're unable to get activated. They're permanently deactivated.
So how come they have different presentations?
The gene is slightly different, but it's the same chromosome. For Prader-Willi, the gene is known as Prader-Willi. It's a different location. And this might be a different gene for Angelman. Okay, it's not the same gene. It's the same chromosome, but it's a different gene. Okay? But the same mechanism is used. The same mechanism is used. So imprinting is used regardless. Malcolm.
That's the same question.
That's nice. Anything else, guys? No questions? Okay, so last thing, multifactorial. Multifactorial means there should be genetics component and there should be environmental. There should be two components to that. Can you think of any disease? Let's talk about diabetes. Let's talk about diabetes because it's the most common and you will be asked about it 10 times, 20 times. Okay, in different settings. Let's say diabetics. In diabetics, you have the beta cells of pancreas produce insulin, correct? Okay. Now, insulin increases what? GLUT transporters. All it does is increases. So insulin acts on other cells to increase production of GLUT, which are glucose transporter, especially 3 and 4. Okay? So that's what insulin does. So if beta cells are normal and you produce these guys are normal, that's good. But now let's do something else. The receptor for GLUT or for insulin receptor. So in order for cells to release or make, in order for cells to make GLUT, that means insulin has to activate them. How does insulin activate cells? Through what receptor? Tyrosine kinase receptor. Very good. So let's say insulin in green attaches to tyrosine kinase. And then tyrosine kinase will increase GLUT. And GLUT will be expressed on the surface of these cells, so now glucose can go in as a channel. Good? Now let's say we have a problem. And the problem is the cell is heterozygous. Heterozygous for this insulin receptor. So they express, let's say normal cell is supposed to express 1000 tyrosine kinase receptor or insulin receptor. Let's say normal cell is supposed to express how many? 1000. But now, if you are hemizygous for that gene, instead of 1000, you can only express how much? 50% of that, which is 500. So already this cell will not be activated to the same potential. You're still okay. You're still okay. But now you start eating diet that is high in sugar, high in salt, and high in lipids, and you do lack of exercise. So you're genetically predisposed. You already have a small number of receptors for insulin. Insulin is fine, not a problem. Receptor is functional, but you have a small number. You know, for insulin to completely have its potential activity, you must have 1000 receptors. But you genetically predisposed, you have 50% less receptors. You're still okay. You can produce some insulin activity, which is 50%. Maybe it's good enough for your body. Now you start eating diet. And you know that low pH does what to this receptor? It causes it to denature. Fatty acid does what? Increases acidity. So fatty diet destroys this receptor. First of all, you have less. You have less. Now you're eating diet. So that was the genetic part, 50% less receptors. The genetic part comes from your diet, from your lack of activity, from your stress, from your anger. All alcoholism, smoking, all of those things contribute to it. Whatever you have left, whatever receptor you have, now they are likely to get more damaged. In addition to that, you can also inactivate tyrosine kinase receptor by phosphorylating which residues? Not tyrosine residue. Threonine residues or histidine residue. Remember I told you that. So don't forget that. Now, when those diet, what they do is they cause phosphorylation of these residues. And this leads to inactivation of the receptor that you already have. And then suddenly you have full-blown diabetes. So there's genetics involved, which predisposes you, makes you sensitive. Now, people who don't have the genetic component. For example, we have two patients. One person A, let's just do this. So to understand multifactorial, number one, you have to know there is genetic predisposition to a disease and there's environmental stress. Okay? Person A and person B. Person A's parents are diabetic. Person B's parents are healthy. They're non-diabetic. Okay? So person A already has genetic predisposition to diabetes. He's more sensitive to the same stress. This person can eat the fatty food all he wants. This person, if he eats it, he's more at risk of developing diabetes. Okay? Does it make sense? So multifactorial diseases, of course, you know, genetic is a huge part of it. But if you restrict your diet, if you stay away from those diets that will put you at risk, you will never have the disease.
Like sickle cell.
Yeah. Not only that, no, glucose 6-phosphate dehydrogenase. Sickle cell is difficult. Still, even you can take a lot of precaution, you will still develop it. It's because upon deoxygenation. So stressful increases that, but even deoxygenation in normal conditions sometimes produce sickling. So what he's saying in sickling, you have, for example, mutated hemoglobin, and this hemoglobin precipitates and becomes clumped together and cause sickling only if you get exposed to increased acidity, infection, and stress and stuff like that. But even sometimes in normal conditions, so it's partially correct. Partially correct. Sickle cell is non-reversible. Diabetes is reversed. Type 2. Type 2 diabetes is completely reversible. You can completely prevent it. So multifactorial diseases you can prevent, and you can do something about it. But autosomal dominant can't do anything about it. Autosomal recessive can't do anything about it. Mitochondrial, you can't do anything about it. X-linked, you can't do anything about it. Pure genetic disease, you can't do anything about it. At least not at this time. There's no gene therapy hasn't been worked. But when it comes to multifactorial, certain multifactorial disease, you can actually reverse it and even cure it. Because it relates on your diet. You're too sensitive. Therefore, you stay away from those things. For example, heartburns. If you're sensitive to acidity, don't take acidic diet. And if you stop taking acidic diet, you won't have heartburns. If you're allergic to penicillin, don't take it. You won't have rash. You won't have tachycardia and so forth. So that's what genetics come from with multifactorial disease. Asthma is multifactorial as well. So if you have, there's pure genetic asthma, and then there is multifactorial. So again, you are at risk of those diseases becoming symptomatic. But if you stay away from all the triggers and toxins and whatever is precipitating that condition, you'll be okay. Okay. Go over these things. Hopefully, you will be okay for tomorrow. But I won't have this on tomorrow's exam. I would only have DNA and RNA. Okay? All right. Let me see if I have